Hello and welcome to my talk about linking mechanistic multi-scale brain simulation, in particular with the virtual brain, together with knowledge technology towards a holistic and better understanding of Alzheimer's disease. I will start directly with declaring that I have no conflicts of interest and introducing the virtual brain. The virtual brain is a neuroinformatics platform that allows personalized and individualized brain simulation based on individual data, and it's based on two main factors. One is empirical data, in particular providing the structural connectivity, so the fibers connecting the brain regions, and the second one is the concept of neural mass modeling. So we bring neural masses in brain regions to life by mathematical models that can simulate neural activity like oscillations, spikes, and typical frequencies. This has been used already to reproduce many different phenomena and healthy brain conditions, and also in several diseases. And it's available as a Python package and also as a GUI. And we are also developing on an ontology describing all the structures within, which I will explain in a bit. Here you can see how simulations with the virtual brain can reproduce empirical patterns as observed in functional connectivity measured with bold functional MRI, just by simulating the activity based on structural connectivity and the underlying mathematics. It is also available as a cloud service, so it's fully usable in eBrains, the platform of the Human Brain Project, and there you can perform simulations on supercomputers and in a GDPR compliant framework. So all the data that is needed for individualized simulations is of course also sensitive data, and here we provide also a framework to protect this data to apply to also European data protection laws. The main idea of why me as a clinician working with the virtual brain is of course to develop applications directly for the use in patients and to improve the treatment of patients. And I'm in particular interested in neurodegeneration and in particular dementia. And here there are three columns that can benefit from research with the virtual brain and brain simulation. First, it's diagnostic, that I will explain how you can use the virtual brain to improve diagnostic processes. But you can also try out virtual therapies, maybe to try out a treatment like a deep brain stimulation in advance, but also maybe to try out new treatment strategies in silico without harming a patient. And the more interesting but also most complex part is to understand the underlying mechanisms. As you can see here, for example, a mechanism in a healthy brain, it's called bistability. So you see the brain switching between an oscillatory and a non-oscillatory state, and by reproducing this in a simulation, we can understand what is behind there. But you also have some cross-sectional approaches that can help into translation into clinics, and that's the field of data availability and making data more available, then the field of knowledge technology, and in general computational methods. So I will start with the first column of improving diagnostics. Here I show you the current classification of Alzheimer's disease as provided by Clifford Jack in his paper, a very famous paper in 2018 in Alzheimer's and Dementia, providing a new framework based on biomarkers for Alzheimer's disease. These biomarkers are amyloid beta and phosphorylated tau, and together they come up with a classification ATN, measuring the pathology of amyloid beta, phosphorylated tau, and neurodegeneration in general, and this allows to classify the full spectrum of Alzheimer's disease and not just one simple thing like saying, okay, that's dementia and probably it's due to Alzheimer's disease, but you can make more precise diagnoses. But this opens also up newer questions, as you can see, for example, in this figure also provided from Clifford Jack in his paper. You don't know how the cascade in Alzheimer's disease is actually done. So the classic idea is the first one, that first amyloid comes, leads to hyperphosphorylation of the tau protein. This leads to neurodegeneration, and then in the end there comes the cognitive decline. But there are also other cascades possible. For example, vice versa, first comes tau, then comes A beta, but also something completely different, that, for example, an independent factor Z produces neurodegeneration and cognitive decline, and as a side phenomenon leads to A beta and tau. All these are possible hypotheses that are explorable if we have good biomarkers. But how to explore these hypotheses? Of course, here simulation comes in as a potential strategy to do so. And I will present you a work that I've done some years ago, where we used amyloid burden measured from PET, positron emission tomography, from ADNI, the Alzheimer's Disease Neuroimaging Initiative. So we took the individual burden of amyloid beta in healthy controls, mild cognitive impairment patients, and Alzheimer's disease patients in each brain region, and translated this individual burden of amyloid beta into an impaired excitability in each region. So the more amyloid beta a region had, the more impairment it was there. And then we combined this with a healthy structural connectivity matrix. So basically we can say we infected a healthy connectome with amyloid beta. And what kind of model we used for that? Here we used the Janssen-Ritt model and implemented the impairment of excitation by an impairment of the inhibitory population. So the Janssen-Ritt model has 3 neural populations, an pyramidal cell population, and excitatory and inhibitory interneurons. And from the literature we know that amyloid beta has a particular toxic effect to the inhibitory interneurons. So these are impaired, and by impaired interneurons that are inhibitory, you get less inhibition, and therefore a phenomenon called disinhibition, so in the end more excitation, and this excitation can again lead into other phenomena like excitotoxicity, so a vicious circle can be started. And we implemented this by changing the inhibitory time constant, one parameter of the Janssen-Ritt model that is responsible for the transduction from the inhibitory interneurons to the pyramidal cells, based on the regional burden of amyloid beta. As you can see here, we just implemented in a simple sigmoid that it will also be demonstrated in the notebook that will be available later in this session. And what we found out is that, of course, the changed time constant leads to a changed response, but it also led to changed spectral properties that were not directly related to the changed signal response function. So basically what we saw is that the Alzheimer's disease patient showed a slowing, particularly a shift from alpha activity to delta activity, as we also know them from empirical data in EEG of Alzheimer's disease patients. And as you can see here, this was a shift that typically occurred in the Alzheimer's disease group, while it was not apparent in the healthy control group and the MCI group, although they also were infected with their own amyloid PET. But of course, they had less burden of amyloid. What you can see here then is that we also could explore the behavior of these models in a spectrum of the parameter G. So the global coupling parameter G scales the connection of large-scale connectivity in these models. So we can explore how the system behaves in an uncoupled state when G is 0, or in a hyper-coupled, hyper-synchronized state, and also everything in between. And that is very interesting, because we can see that besides the spectral differences in a particular fitted range of G, for example, the most realistic signals were in these low areas of G, but we also see that by bringing the system into a different regime, we can see differences between the diagnostic groups. So the healthy control patients had a different bifurcation here, so where the system changed, for example, to the non-oscillatory change, than it was the case for Alzheimer's disease patients. And we actually explored this further to analyze the potential to improve the diagnostic process, which I will show in the following. But first, we also tried out virtual therapy. So actually what we did is, because our model introduced artificial hyperexcitation into the system, we tried out a treatment that acts against hyperexcitation, which is memantine, an NMDA antagonist. And what you can see here is that indeed injecting virtual memantine by reducing the NMDAergic, or let's say the glutamatergic transmission in the system, reversed the effect that was introduced by the amyloid beta. And as I said, we also explored how this can be used for a diagnostic process. And if we have here a look to the left, we can see how the regions of the brain regions of the three different groups have different values in these three dimensions. So we have the PET value for tau PET, for amyloid PET, and the artificial, the simulated frequency, how they were oscillating in. And as we can see, there are particular differences between these three groups, but they are kind of convoluted, so not so suitable for a classic biomarker approach, just saying that there's a cutoff value and everything above this value is pathologic or not. But of course, it's a good approach. It looks like promising for using machine learning. And therefore, we employed a machine learning model based on support vector machines and random forests, and trained this first only with the empirical features, which were in that case amyloid PET, tau PET, and MRI volumetry, and then only with the simulated features that were the local field potential frequencies, and then all together. And what we could observe is that there was a significant benefit of the combined approach. So although we did not use more data, we just used the simulation of the same data, but the simulation provided kind of hidden parameters that helped to improve the diagnostic classification by about 0.1 in the rated F1 score. And that was particularly observable for the combined feature set that showed an improved classification of the mild cognitive impairment group in the study. And we also found out that the regions were actually very plausible, which is not always the case in a machine learning study, of course. So we could see that the parameters that were most distinctive for this decision of the machine learning model were, for example, the temporomesial amyloid beta values, or the entorhinal tau values, or the frequencies in the subcortical regions where typically also rhythm generators take place. So to summarize, we can say that it's possible to improve diagnostics by augmenting machine learning with simulated data from a large-scale brain simulation. It's also possible to translate empirical into mechanistic biomarkers and to integrate multiple modalities into one multi-scale brain model. So, as I've shown you, we can combine many different modalities, as for example here the different PET values, structural and functional MRI, and in the end come up with a parameter that holds larger potential to augment the diagnostics than the single modalities before. So the second column will now be to how to make use of knowledge technology to understand the underlying mechanisms. The integration of knowledge in science is, of course, a crucial task. And here we can make use of the concept of knowledge graphs, or in general of graphs and ontologies. And how could this be the case? So first we start with the data, and modern databases often are already organized in the form of a knowledge graph. So for example, eBrains, the database of the Human Brain Project, contains a knowledge graph that describes all the data that is stored in this database and makes it more accessible so that you can actually get access to, in particular, the data that you are looking for and that you are needing for your research question. Then we also designed something like this for the mathematics of the virtual brain, which is actually the virtual brain ontology that was developed by our team to describe the mathematics inside of the virtual brain and of the different parameters to compare the different models to each other and to help researchers to design cause and effect models as I have described them before. And then the last thing is that there are also knowledge graphs available describing biological pathways and mechanisms. For example, they are available for proteins, protein interactions, but also for particular diseases, as for example for Alzheimer's disease. And this is a huge resource to systematically access biological knowledge to implement it in a brain simulation. But how can we integrate this biological knowledge into a brain simulation? Because when we know that protein 1 interacts with protein 2, it does not help us to set up a simulation if we don't have suitable data here. And here we come up with another tool that was developed by our team, still unpublished, but will be published very soon. And that exactly helps to find this localization. So here you see, for example, a knowledge graph describing the protein interactions and also interactions with other chemical compounds in Alzheimer's disease. And we want to know where particular in the brain these interactions take place. And this can be done with a tool that we call the virtual brain adapter of semantics. And it does actually what the name promises. It adapts semantics and creates new data out of a semantic query. And how can this be done? Basically, we make use of an existing technology that is called Skyview, which is a search engine similar to PubMed. It contains the same scientific articles as PubMed, but they were text mined. So basically what was done is that all the text of these articles were mined with an algorithm, and particular concepts were marked as these concepts. For example, genes were marked as genes, and chemical compounds were marked as compounds, biological pathways, and also anatomical terms. So we can find and we can measure how often an anatomical term is mentioned in a particular context. And this allows us to extract then statistics about the appearance of these anatomical terms for a particular search query, as for example for Alzheimer's disease. And we can find where Alzheimer's disease is associated with in the brain by extracting and interpreting these statistics. And how this is done? This can be done because we developed a unique transfer matrix. So we manually aligned about 1,000 anatomical terms to a three-dimensional template of the brain. So for example, we assigned that, for example, the term frontal lobe belongs to particular parcels of the brain. And here we made use of the parcellation of the Human Connectome project, also known as the Glasser parcellation, with 180 parcels per hemisphere in the cortex. And in addition, we used the subcortical parcellation from free surfers, the so-called Fischl parcellation. And this will be available also as a Python package, but also as a GUI. And you can then directly search for a particular concept. For example, also something very simple like memory, but it can also be a gene or whatever you want to. You can also search for free texts, and you will get a map of the relevance, the association with this search query with the particular brain area. So you can extract a map, an artificial data modality out of a search query. And this helps to improve brain simulations in future, because we can add information about processes, actually about any possible processes that we can query for by injecting these maps into the simulation. But first we have used it to interpret empirical data. And this is a study that is also unpublished, but I still want to show it to you, the preliminary results. So colleagues from us provided us with patterns of volume changes in a population at risk of developing psychosis, and these volume changes were predictive for the onset of psychosis. And we wanted to analyze what mechanisms could stand behind these volume changes. And for that, we used support vector regression. So we just picked genes from the whole genome and tried to reconstruct the pattern of volume changes that was identified by a machine learning, actually also support vector machine ensemble, that identified the brain regions that are predictive for psychosis. And we found out which gene maps can together best explain this pattern. And that is kind of a very new and unique approach, and led us to, in the end, to three main clusters of processes, which were the neurotransmitter and amino acid homeostasis. Of course, not so surprising, because it contains, for example, the dopamine and the other catecholamine homeostasis. But it also included neural growth, for example, with a brain-derived neural growth factor, and a very interesting a cluster about interleukins and tumor necrosis factors, so immunological factors, which is of course very interesting, because this is a cluster that we can possibly also treat in an experimental study. But to make this, you can of course do it like I've done it before and showed you for Alzheimer's disease, but you can also of course use our new technology, the virtual brain ontology, to describe this and to develop a new model here, and to define the mathematics. And how can TVBO help here? So basically, as I said, TVBO describes the mathematical relationship between the particular parameters, and it's itself a graph. So it's an ontology, so a graph with some more annotations, and the entities of the graph are connected to each other, but also connected to biological concepts. So here we relied mainly on Go, the gene ontology that describes biological processes. So you can find out, for example, which parameters throughout all the different models of TVB are connected to, let's say, inhibition, or for example to NMDA receptors, or to impaired signaling of a particular receptor. So in general, your creativity has no borders to use this, and because it's standardized and it's a highly annotated framework, it also makes the choice of such a mechanistic model more transparent and also more reproducible. And just to show you how this can be done, for example, can be used all together, I can show you how we reproduce the result that I showed you in the beginning about Alzheimer's disease with slowing in Alzheimer's patients based on A-beta, and this can be reproduced, for example, if we use artificial amyloid maps by searching with TV-base for amyloid and Alzheimer's disease, and in comparison we search for amyloid with other diseases, for example frontotemporal dementia or Parkinson's disease, and there we don't see then this slowing. So to summarize how to use knowledge, technology and mechanisms to improve the research for Alzheimer's in brain simulation, we can make use of existing knowledge that is available in the literature, in databases, and in existing knowledge graphs. We can make them available in a better and more richly in an annotated form, and we can combine different data modalities together with computational modeling. So in the last part, I will shortly speak about how to develop new treatments based on this. And here for that, I again come back to TV-base and show you what we also did by exploring a biological knowledge graph of Alzheimer's disease. So this is a knowledge, this was this vicious circle that you see on the right was identified by finding out the most informative maps of a set of concepts in an Alzheimer's disease associated knowledge graph, which is called Neuroinformatics. And these concepts were linked together by the edges that are in the original knowledge graph. And as you can see, for Alzheimer's disease, they built this particular circle, which is of excitotoxicity. As you can see, that amyloid beta leads to calcium influx, and this then leads to some assembly of neurofibrillary tangles. So it's basically tau protein. This leads to an inflammatory response, and this inflammatory response leads to an activation of NMDA receptors and an inhibition of GABA receptors, which then in the end also leads to more calcium, and the circle is started. So just without touching patients or acquiring new data, just by the abstract analysis of existing knowledge in the literature, we can reconstruct such a mechanistic circle that can in general explain a lot of things that we can observe in Alzheimer's disease. And it can, if we analyze the circle further, we see that there are three main drivers of the circle, which is the tau deposition, the inflammation, and hyperexcitation. And this is, of course, something that can be targeted for a potential treatment. So, for example, tau is already, there are already studies with anti-tau vaccines in the field that try to filter out phosphorylated tau proteins from the brain. But of course, neuroinflammation can be addressed by immunomodulatory treatment, and hyperexcitation can be addressed with a clever form of stimulation, can be invasive stimulation as DBS, but it can also be non-invasive stimulation as, for example, TMS, where there are already some applications in Alzheimer's disease. Here we see how to explore the effect of a stimulation on the brain. That's the work together with some colleagues, mainly done by Jill Meyer, where we see the effects of virtual deep brain stimulation in Parkinson's disease. And that's a complex model where TVB simulation is in the cortex is combined with a spiking oscillator model in the basal ganglia. And what we can see is that by stimulating the subthalamic nucleus, as it is done in clinical context for Parkinson's disease, we see an effect on the prefrontal supplementary motor regions as we know it, and also we clinically see it, and we also see an activation of the thalamus, in particular for the subthalamic stimulation that helps to again start the movement. So actually we can reproduce the effects that we would expect from empirical data, again by just in silico applying a stimulus to an artificial brain. So to summarize the treatment column, I would say that the identification of novel targets is probably the most promising but also most complicated fields. We can then try out these treatments either in a pharmacological simulation but also with stimulation. And we can also, of course, improve the stimulation further, for example, by using the simulation to find the individualized pattern of stimulation, ideally maybe also in a closed loop system in the future. And this can, of course, also maybe in the future be combined with wearables and digital applications to make it even more accessible. So in the end of my talk, I will just give a short outlook of what can further be done in this field. So the diagnostic approaches can, of course, be extended to other disease contexts and to other simulations with even more data. And of course, you can also try out other classifiers. So here in the example that I showed you, we used a conventional classifier, support vector machine. The more data you have, then also, of course, deep learning approaches make more and more sense. For virtual therapy, we can even go either more into the microscopic area by refining pharmacodynamics, maybe implementing the effects of particular receptors or particular ion channels, but we can also go even more into the creative approach and try, for example, out new stimulation target, thing that is actually not doable by ethical reasons so easy in real patients, but in simulation you can of course just try out what happens if I stimulate not the subthalamic nucleus but, for example, the fornix, and you can just see if this is maybe something that is worth to have a closer look in empirical data. You can also make use of control theory to fine-tune the stimulation, as I said before. And finally, speaking again about mechanisms, what I had not much time to talk about today is that you can, of course, use a lot of mathematical tools to analyze these mechanisms further. So there's the bifurcation theory, so you can have a look into the bifurcation diagrams to understand why a system changes between one regime to another. And you can also in that field integrate molecular data, for example, on ion channels, as I said before. And altogether, I have the vision in the future that we can really bring all these technology aspects together to, in the end, also design clinical studies, clinical trials with interventions where you have all the other parts also included, because I think this would really facilitate the research, especially on dementia, when you have a clinical study, but at the same time acquiring high quality data. You store this data in a responsible and accessible way so that it can be used also for other analysis, and at the same time you use the state-of-the-art technology of brain simulation and integrate also molecular and multi-omics data into these simulations to, in the same time, analyze and understand, for example, what happens in the studies, what happens with these patients in a clinical study, and maybe why some patients benefit more than others. And I think this would be a perfect fit between computational neuroscience and clinical science in a really translational context for the future. And with this, I end my talk, and I thank all people from my team, and hope that you learn something about brain simulation of Alzheimer's. Thank you.